So this is a growing toy. It's a toy that grows exponentially after being submerged in water. It takes 5 days or so to grow into that. Would you believe me if I said that this grows faster than certain types of tumors? That's kind of insane. Like, it takes 5 to 10 years after a cell first becomes cancerous to turn into a detectable invisible tumor. That's 5 to 10 years of you not knowing that you were a host to cancer. That's a significant portion of someone's lifetime and is absolutely insane. 2 months ago, my grandfather visited a doctor. The doctor diagnosed him with tuberculosis, but he didn't have tuberculosis. It was cancer. Doctors often think, and patients often think, it's really just hard to detect cancer. Cancer tends to hide from us, from doctors, and from our own immune system. But there's one thing that it can't hide from: our genes. Let's take a look at what the company Freenome's doing, for example. They take routine samples for a blood, and then they sequence them to detect genetic variation and to see whether we have a high or low chance of getting cancer in the future. This takes care of half the job, detection. But what about prevention? There are other things like CAR T-cell therapies as well. CAR T-cells are essentially the biggest and baddest in our immune system. They go out and kill. But when it comes to cancer, they're not too effective at that. That's where CARs come in. They're chimeric antigen receptors. So what they're essentially doing is that they're boosting T-cells. They're making them bigger and badder, and they can kill cancer. But there is no silver bullet. There is no one single cure to cure cancer. So we have to develop many of them and layer them one on top of the other. So here's what my proposition is. In essence, it's the p53 gene inside of an adeno-associated virus, which is then put inside of a DNA nanovirus. Okay, so that was a lot to unpack, but here's what it is broken down. So the crazy thing about p53 is that it's actually mutated in more than 50% of all cancer patients. That really shows how significant it is to our cell cycle. The way that it works is basically it can either do one of two things when it detects genetic variation or predisposition. Either one, apoptosis, which kills the cell, or two, cell repair, which repairs the cell and makes everything better. I like to think of p53 as a sort of bounce. It guards cells from doing mitosis. If you're mutated and cancerous, you can't do mitosis and you can't multiply. But if you're completely fine, mitosis is that one. Next, you have retroviruses. They're essentially this type of RNA virus that can go and insert its own genome into the cell that it invades. Viral vectors, on the other hand, are this type of retrovirus in which the virus DNA is actually taken out of the thing, and inserted is the gene that's functional. To get a good understanding of the application of this, we can take a good look at the company Bluebird Bio and what they're doing with this type of technology. They're trying to cure sickle cell disease by taking a gene that's commonly mutated in sickle cell patients and then putting it in a viral vector. This is an adeno-associated virus. It's this special type of viral vector that doesn't incite much of an immune response. Also, it doesn't, unlike many other viruses, it doesn't hurt us. So this really is the way to go. Next up, you have the Mona Lisa. But it's not the Mona Lisa. It's the Mona Lisa in our DNA. How cool is that? Essentially, they synthesize these DNA strands and make them into a image or a structure. But it doesn't have to only be used for two-dimensional structures per se. This right here is a DNA nanobots. It's currently being used to carry certain drugs through the bloodstream at a nanoscale. It's super small. It works by this thing called an aptamer. It's this lock that opens up when it detects a certain protein. And specifically in the case of p53, if it detects certain proteins that are overexpressed in cancer, then it'll open up and release the retrovirus. So here's my idea once again. It's the p53 gene inserted inside of the retrovirus, which is the adeno-associated virus, which is then put inside of a DNA nanobots. This is really cool for 3 main reasons. These being its detection, therapy, and suppression all at the same time in this tiny little package. using Freenose technology or CAR T cells, or you're layering them all on top of another. It just isn't worth it to wait 5 to 10 years to find out you have cancer.